Hi, I'm James. Really nice to meet all of you and look forward to talking with each of you more during the rest of the week. So I'm a faculty at Stanford, and today excited to tell you a bit about clinical trials and also about, in particular, about how we can use this idea of in silico clinical trials, right, to help us design new clinical trials and also help us to improve this precision oncology. So many of you probably know that these clinical trials is really the gatekeeper and bottleneck of all of medicine, right? If you want to get these COVID vaccines, right, they have to go through the clinical trial process first, right? And these trials actually have a surprisingly long history, right? So this is actually the first example of a clinical trial I found that's written down in the written record, right? So it goes all the way back to 600 BC, right? So it's over 2,000 years ago. And the first recorded trial actually is recorded in the book of Daniel. And it's an interesting story, right? So the story here is that the king of Babylon, King Nebuchadnezzar, had the idea that if his citizens drink more wine and eat more meat, that will make them healthier, right? So then he had a decree that everybody can only drink wine and only eat meat. Of course, there are some people, some of the citizens were not so happy about that because they want to drink water and eat some vegetables. So they complained, and then the king decided that, okay, let's actually have an experiment. So basically, for the course of about 10 days, right, so he had two groups of citizens. One group would follow the king's diet of wine and meat. The other group would follow their normal diet, maybe of some water and vegetables. And after 10 days, the king had some people go around and actually measure the health status of each of these participants. And much to his dismay, the water drinking vegetable eating group actually looked healthier, right? So then he had to roll back that decree. Yeah.
Were they randomized?
They were not randomized. So yes, so the ideas of these clinical trials, right, and we'll actually come back to randomization a bit, so that's a good point. So of course, the clinical trials have changed quite a lot, right, over the last 2 millennia. But this core idea, right, of having this relatively well-controlled settings, right, with a well-controlled outcome of measuring the effects of a particular intervention on the health outcome of humans, right? So that's sort of a common thread that's been persistent across these 2,600 years. Of course, the modern clinical trials have gotten much more complicated and more expensive, right? And you can start with like hundreds of therapeutic drug candidates, right? And only one of them will actually make it to the patients by going through these different stages of the clinical trial process. Right? Each trial itself can also be hugely complicated, right? It could cost on the orders of hundreds of millions of dollars to run just to conduct each trial. And also accounting for the fact that so many of drugs fail to get through the different trials, right? So the average cost of actually bringing in a drug actually onto patients is typically on the orders of, on average, several billions of dollars. Right, so that sort of motivates the things that we want to discuss today, right? Is that these trials are actually very expensive, right? So and certainly very important for the entire biomedical translation pipeline. So basically, anything that we can do to make this process maybe more efficient, then that can potentially have a huge impact on all the different steps of this pipeline. So that's the motivation, right? And in particular, today I want to tell you a bit more about this idea of these in silico clinical trials. Right, so this will be a mixture of sort of a research talk and tutorial. And in particular, we'll talk about in silico clinical trials that we can conduct computationally, right, using real-world clinical genomics data. So real-world here means that the data is actually coming from actual patients who take these treatments, not from patients who are enrolled in these specialized clinical trials. Right, clinical genomics data, here we're referring to basically data that come from medical records and from genomic testing that people have done on these patients, for example, with Foundation Medicine tests for cancer genomics. And I'll tell you sort of how these things work, and I want to sort of illustrate this by first giving you sort of some motivating challenges with existing randomized clinical trials, right? So to motivate why it's useful to think about these in silico trials. And I want to tell you about sort of 2 example applications that we worked on recently, right? One's on using these in silico trials to make the actual trials more efficient and more inclusive, more diverse. And the second is to use these in silico trials to discover a large number of predictive biomarkers for cancer treatment recommendations. And in the last part, I will go back and discuss a bit more some of the best practices from a statistical side, data science perspective, of how to do this kind of analysis. Okay. So we'll start with the first setting. I also want to tell you a bit. So most of what I've described here was described in this paper that we published last year, where we developed this computational platform called Trial Pathfinder. So I'll explain what that does. That uses this real-world data and these in silico trials to help us to design actual clinical trials. And we're very excited that this actually platform is now being used by Roche and Genentech and others to help to guide the designs of their new cancer oncology trials. And this work is led by Rixiang Liu, who's a fantastic PhD student at Stanford working in my group. So first, I want to give you some more context of how do people design clinical trials and what are the key challenges. Right? If you think about the design of a clinical trial, it's actually a really complicated object, right? And one big factor of why it's so complicated is that each trial has to decide which patients are eligible to participate, to enroll in that trial. So if you look at the trial protocol, it will have many pages of PDF, right? Each PDF, right, will have lines, and each line will lay out different rules for excluding patients that says which patients are not eligible to participate. Right? For example, if patients are too young or too old, or if their biomarkers outside of some range, or if they have previous diagnosis or previous treatment, oftentimes they're excluded from participating in these trials. So it's becoming recognized, right, that these trials are actually oftentimes very restrictive, right? And that has several consequences. It actually makes it could be quite harmful for the patients, because in cancer, most of the cancer patients actually would like to participate in cancer clinical trials because that's how they can get access to some of the latest treatments, immunotherapy, for example. But many of them are not eligible to participate. The second consequence is that it actually harms the drug developers. Right? So a large number of the cancer trials, for example, fail because they fail to reach enroll the minimum number of patients, just because it's actually so hard to enroll patients that satisfy this eligibility criteria. And third point, maybe the most important, is that actually it's really bad for all of us. Right? So the clinical trials, because they're very restrictive, the patients that enroll in those trials are often called the Olympic athletes of patients. Right? And how well a treatment works on these Olympic athletes, right, may not reflect how well the treatment works on regular people like us. Right? So there's a huge distribution shift. And people are starting to recognize that the results from clinical trials, right, even these randomized, well-controlled trials, often do not generalize to the general population. Right, so for all of those reasons, right, we thought it's actually important to look at, can we help to guide the designs of these trials to make them maybe more efficient and more inclusive. So the question you might have is, okay, so how do people currently design these trials? Right? How do they design these eligibility criteria? When you think about what patients to enroll in a particular trial, there are multiple objectives, right, they often try to optimize for. So certainly you want to make sure that the patients that enroll in the trials would take these treatments and not have adverse events. Right? There's the safety of the patients, which is paramount. And condition on things being safe, right, you also want to enroll the patients that somehow effectively highlight the treatment effects, right, the effect of the drug that you're trying to test, because otherwise then if you don't show a treatment effect, then it's hard to get it through the FDA process. And unfortunately, the current designs of these trials, it's often very anecdotal, right? Very heuristic-based, because we don't really have any good data-driven tools to help us to design this eligibility criteria of which patients to enroll to recruit for a particular trial. Just to illustrate this, I'm showing you this a very particular concrete example. So I just took some screenshots of an actual trial that some of our collaborators are designing. Right? This is for a particular it's a draft protocol for a particular treatment for lung cancer. And the first thing that people often do is that when they're designing a new trial, they actually just go back to their database and find and copy and paste over what is the previous trial that the pharma company had done that's the most similar. Right? So they find that protocol that they had done before, and they'll copy and paste that over. So that's the starting point here. Right? And then they would just go through line by line and see, okay, do you make any modifications to the patient population? So each row here actually corresponds to one rule for excluding patients. right? For example, the first one that I highlighted here is looking at this thing called neutrophil count, right? Which is a common biomarker that's used to exclude patients from participating in these trials. So the details are not so important, but each row corresponds to one rule for excluding patients. And you can actually see this really interesting debate that happens in the sidebar of the Google Doc, which is captured here, right? So the initial trial that's copy and paste over had this neutrophil count that's pretty restrictive, right? So somebody asked in the Google Doc, okay, can you relax this from 1,500 to 1,000 to enroll more patients? And a third person would chime in and say, oh, we don't really have any data to decide one way or the other to set the threshold. Another person would say, okay, let's then just keep it to be what was done before, just to be consistent and to be more conservative. Right, and then for the next one, there'll be a similar discussion, right? So people would say, okay, can you actually relax the hemoglobin threshold to enroll more patients? Right, and there'll be another debate that says, okay, we don't have enough evidence, let's just keep it to be what was done before. So that's actually the current practice. Right, so the upshot of this, right, is that because these trial designs are often quite heuristic, based on the previous history of a particular team, right, that different teams end up using these very wildly different rules for deciding what patients to enroll in a particular trial. And to just visualize this, I'm showing you here sort of 10 relatively recent clinical trials looking at drugs for advanced non-small cell lung cancer. It's the most common cancer type. Each row corresponds to one of these trials. Each column corresponds to one of the rules that's commonly used to actually exclude or include patients. Right, so some set of these rules, for example, in the beginning corresponds to different biomarkers or previous diagnosis or different lab values, right, for excluding patients. And I color-coded each square to be yellow, right, if that particular trial used that rule to exclude patients. Otherwise, it's colored gray. Right. The main point here, the takeaway here is just even visually, you can see that even among trials that are looking at very similar drugs, the same cancer type, they just end up using very different rules to decide what patient cohort to recruit for that trial. Right. And this is sort of recognized to be really a fundamental problem, right? So for example, like the National Cancer Institute in the US have put out mandates, right, that basically all of the cancer trials needs to be simplified and standardized, right, in order to enroll more patients and more diverse patients, right? But really how to do this is very challenging. That's just where we want to use these in silico trials to help. Okay, I'll take a pause here to see if the problem setup makes sense to folks. Any questions? Yes?
Sorry, maybe this is a silly question, but what exactly do you mean by an in silico trial?
Yes, I'll set that on the next slide. But basically, the idea is use use data from the medical records to run a clinical trial on the computer rather than in the real world. Yeah.
Is there data available from past clinical trials on what the criteria was and how many patients were?
Yes. Yeah, I'll also show that. We'll basically compare the data from the past trials with the data from the trials given our designs. Yeah. Okay. Sounds good. Okay, so then let's dive into the methods a bit more, right? As computer scientists, right? So really the core problem here is really a problem of counterfactuals, right? I have all these patients who want to participate in the clinical trial, but they're excluded, right? They're not eligible to participate. What I really want to know is which of these patients who are excluded would have benefited from these treatment that are tested in the trial safely, right? So that's really the core of the problem. And idea here is that, okay, so how do I really test that counterfactual, right? So here's the idea, right? It turns out that after the drugs, right, comes on the market, after it's approved and it comes on the market, then a lot more people take those drugs, right? A lot more people take those cancer treatments, even if those patients were not eligible to participate in the original clinical trial, right? So even if clinical trial is recruiting and tested on these Olympic athletes of the patients, right, a lot of the regular patients still ends up taking the treatment after they come online, right? So that motivates the idea that, okay, so actually, can we just go into a large number of medical records, right, to find those patients and see what actually happened to them, right? And to do this, we collaborate and we work together with the Flatiron Network, which is maybe the largest network of cancer patients. It has maybe 280 cancer clinics in the US, about over 2 million patients. And what's really nice is that they actually spent many hundreds of millions of dollars to curate these medical records, to harmonize them. Right? So for each patient, we follow them over many years, so we know what treatment they're given, their lab values, and their outcomes. So we're going to use this data to do these in silico trials. Yes, please, go ahead.
So that's sort of the way you're talking, there seems to be an assumption that the clinical trial is beneficial, right? But when you're running a trial, you don't know. Right? And so
Yeah.
there seems to be some bias because the data you're looking at are looking at the successful ones. And the non-successful ones, you don't have data. You know what I mean? How do you deal with that?
Yeah, so we're going to look at, this is a good question. So we're going to look at a bunch of trial protocols, including the ones that are not successful, right? So we have that data as well. Right? And what's interesting is that even many of the drugs that come on the market, yes, then they have to be successful for some indications, right? But to the same drugs, for many of the other indications, they actually correspond to failed clinical trials. Right? So we'll have that information to compare with. Okay, so here's how the idea works of what we're calling these in silico trials, right? So it's actually very intuitive. So the idea here is that, okay, so we have a trial that we want to basically computationally emulate, right, on my computer, right? So the trial, right, has these components, but the most important ones, then you have some treatment drug, maybe your immunotherapy, right? You have some control, which could be your chemotherapy, and you have a set of rules for deciding which patients are eligible to participate. Right. In a simplified example, I'm showing you like maybe one rule is you want the bilirubin to be less than some threshold. So bilirubin is one of the commonly used lab tests to assess for liver function. Right. Okay, so in this toy example, so what we're going to do is that we're just going to these medical records, right, of these 280 cancer clinics in the US. We can look for patients in the records who actually took the same treatment drug, right, took that treatment, right, so that generates and satisfies this threshold of bilirubin, so it gives us this one digital cohort, right? I can also look for all the patients in the medical records who took the control treatment, right, who also satisfy the threshold, that give us like another digital cohort, right? These two digital cohorts may look very different, right? Maybe they're taking these things at different times, where they have different demographics. And then it's at the high level, we sort of want to match these distributions, right, so that they're comparable in terms of their baseline demographics and covariates. Right, and just as a side, and we'll come back to this more in the third part. But there's a bunch of statistical ways we can try to match these distributions. Right, you can either use more like propensity score ways or using machine learning methods. And we have some papers on using more like optimal transport for matching these different data, these digital cohorts. But the main point here is that the goal of these matching these cohorts, right, digital cohorts, is that so that they're comparable at baseline, right? They have similar kind of average, aggregate demographics and treatments and previous comorbidities, right? And the reason why that's useful is now you can actually compare them, right? Since we also have the longitudinal outcome of those patients and their adverse events, right? We can actually estimate things like hazard ratio, which is sort of a common metric to see like what are some of the adverse events or survival, mortality, or death events of these patients. So the power of this approach is that because it's all done in silico, right? So I can just very easily computationally change these different thresholds, right? Instead of setting bilirubin less than 1, which is maybe restrictive, I can broaden that, right? And by broadening it, the threshold, I enroll more patients. I find more patients in my medical records that satisfies these, that can then go into my digital cohorts, right? I can match their distribution again using the subroutine, right? And I can also estimate the outcomes, right? Using this now with corresponding to these larger cohorts. And the point here is that if when I compare the outcomes of these patients using these more relaxed cohorts, right, I don't see more adverse events or I don't see worse survival outcomes compared to the more restrictive conditions, right? That gives me some data-driven evidence, right, that I could potentially relax that threshold, right, to make it to enroll more patients in that trial. Yes.
I have a question
Yeah.
Speak to whether these are like manually curated for, things that can be tough to figure out when people started the drug, if they stop, what sort of side effects. How did you get that for the EHR, and was that
Yeah.
process?
Yes, so it's a good question. So I would say it's probably like 95% manual and 5% algorithms. So they do have a network of like actually thousands of clinicians who go through these medical records from all these different sites to extract like progression information, survival information, adverse events, and to harmonize them. So they have some NLP side, and we actually are working with them on some of the NLP things. And I should mention that for disclosure, I'm on the SAB of the board on Flatiron to help them on the NLP side. So we do use some, they are using some NLP algorithms, but most of these validation and harmonizations using manual curation. Yes?
So the match distribution, maybe I missed that, but that's so that you have to avoid the systemic bias, or?
That's right. Yeah, basically you just want to avoid any potential biases to remove potential confounders.
And so that's typically, I mean, from my experience, there's a lot of noise over there, right? So it's not going to be a very, especially if you look at real data, EHR data, then it's going to be, it's pretty hard to say that you're going to get the same population, right?
Yeah.
So how do you deal with this?
Yeah, so here, it's a good question. So, so here, I guess we really benefit from here. The EHR is actually very well curated, right? So you have the longitudinal outcomes of each patient, right? So we know we have relatively complete data on exactly when they're given each treatment, when did they progress onto the next line treatment, and when did they die, where the death information is matched from Social Security records. And we also have the lab values and ECOGs for each of those patients and their staging. So it's relatively well curated, so that enables us to try to control for as many of these confounders as possible. But you're right in that this is there's still potential limitations, which is why I'll mention how we actually use data from randomized clinical trials to try to validate all of these results. Yeah, good questions. Okay, so here are maybe some other questions that you might be thinking about. One big question, if I were you, is that okay, so all these ideas we mentioned just now, right? So it's looking at drugs through the EHR, but the drugs have to be on the market, right? But what if the drug is actually not on the market, right? So what if you're actually running a clinical trial for a new drug? Then that information is maybe not in the medical records. So what do we do there? So it actually turns out that many of the new clinical trials are actually testing for older drugs, right? Even like, for example, maybe you have like an immunotherapy that's approved 5 years ago, right? There's still many new trials that are testing that immunotherapy for different variations, for different endpoints, or for different diseases, right? So actually, most of the new trials are actually still testing for old drugs that are already on the market in some other form. And second is that even if you have a new drug, right, there are often very similar drugs with similar mechanisms of action that's already approved, right? You can have like another new immunotherapy, right? Maybe another checkpoint inhibitor, but there are a bunch of other checkpoint inhibitors already on the market, right? So you can try to use that information to help us to do this analysis. Right, so second big question is also, I think, the question that Iran had is often that come up is, okay, so this is looking at observational data, right? So there are always potential unanswered confounders in these observation data, right? So that is always a limitation with any kind of analysis like this. As we mentioned, we really benefit from having very highly detailed EHR that's very clean and curated. But ultimately, we will and we need to validate these results using data from randomized studies. Okay, so then just to summarize this in silico methodology, right, for the trial Pathfinder, right? So you start with the actual trial, right? So it's written down in these 100-page of PDF, right, in natural language. You can do some NLP to extract the relevant rules for including or excluding patients, right? So once you get those rules, then you go into these large number of different medical records to identify to build these digital cohorts, right? We can match them to remove confounders, and then do the survival analysis to estimate like hazards and estimate adverse events and outcomes. And the point here is that we don't just do this once, right? We can actually do this millions of times in parallel because it's all in silico. Right? Each time we do this in parallel, we're actually testing for a different combination of eligibility rules, right? I can change my thresholds, right? And the reason why I want to run so many of them in parallel, right, is because there could be different interactions between different eligibility rules, right? So I want to test a lot of these different combination of them. Each time, each combination corresponds to one of these in silico trials. And at the very end, right, I can sort of summarize all of these learnings from these millions of in silico trials simulations into one set of data-driven recommendations for the design of these trials. Right? Yeah.
Isn't there some risk of kind of overfitting, of kind of nitpicking, which is not what you want? You want to actually widen, right, the criteria of somehow kind of narrowing it to get kind of an optimized, I don't know, difference between
Yeah, so
case and control.
it's a good question. So here we do show that in general we do widen the criteria. Yeah.
Is that because the statistical power is increased? And
Yes. Yeah. Okay, so here's just one example of how this looks like. Right. So so again, we look at this bilirubin. So each, we have these 10 advanced non-small cell lung cancer trials. So each curve actually corresponds to one trial, right? So originally they all had maybe different thresholds based on bilirubin for excluding patients, right? And then each curve corresponds to the in silico or the counterfactual version of that particular trial, right? Where we can actually relax the bilirubin thresholds, right? From the left side is more restrictive, and the right-hand side is less restrictive. And we can see, okay, if I relax that threshold, how does that actually affect the outcomes, which is the y-axis, that's in terms of the hazard ratio, right? So lower the hazard ratio, the better it is. So the main takeaway here is that if you actually look across these trials, there's a range, right, of this bilirubin value, right, at least according to the counterfactual analysis, where we could relax this threshold to make them more inclusive and less restrictive without making the hazard ratio change too much or without increasing the hazard ratio. Right, so that's sort of, then this will be an example where we can actually provide some data-driven recommendation of how we can set these thresholds. And you can do this systematically, right? So if you recall, like this original table of all these different criteria that people use, there are all these different columns here, right? For each of those, we come up with do a similar analysis to provide a data-driven recommendation of which ones to use. And the upshot of this, right? So we applied this to a bunch of different cancer types. Here I'm showing the results from lung cancer. We also applied it to melanoma and breast cancer and others. So here's like 10 lung cancer trials that people had actually run, right? And then the first 3 columns corresponds to if you actually run the original trials using the original criteria, right? Here's how many patients are eligible and what's the hazard ratio of those patients. Right? And the last 3 columns corresponds to now if we actually adopt this data-driven recommendations, right, from this algorithm, from looking at these large number of in silico trials, we show that we can actually, on average, more than double the number of eligible patients while still actually reducing the hazard ratios, right? So this actually means that, again, hazard ratio lower is better, right? So this means that many of these patients that were systematically excluded before, right, now that they're eligible to participate in the trials using our broader, more data-driven eligibility rules, and those patients would actually have benefited from this treatment to a similar or even greater extent as the more the Olympic athletes that were eligible in the original trial. And really quite critically, right, so by taking this data-driven criteria designed to enroll more patients, we actually are enrolling a lot more women, more minorities, and more older patients. And many of those patients were actually systematically biased or excluded from participating in clinical trials. Now, by taking this data-driven criteria, many of them are eligible to participate. Yeah.
Why does the make 17 have a higher odds ratio than, or hazard ratio in the second one?
Oh, you're looking. Yeah. So I think there could be some specific reason for that trial. Yeah, we can discuss that more. Okay. Each of these trials also have a bunch of their subtleties involved. Oh, yeah.
Was there like a specific criteria that you found more often for these patients, minority women, older patients, et cetera, out of trials?
Yeah, it's actually a lot of the lab values, right? So a lot of these trials ends up being pretty conservative in terms of like the lab values, like hemoglobin and so on, have to be within a particular range, right? And the sort of indirect effect of that is actually excluding a lot of patients. Yeah.
So I might have kind of missed something. So if there's a new drug and somebody asks you what should be the eligibility criteria, do you do some kind of representation learning to see which old drug it's similar to, or how do you do it?
That's right. Yeah. So if it's really a brand new drug, right? First, we will see, okay, like are there some nearest neighbors, right, of similar mechanism of action, maybe like another checkpoint inhibitor that we can use, right? Then we can that's already approved. And then we can use the recommendation that we learned from those previous drugs to try to extrapolate a little bit to the new one. Yeah.
You have smaller standard errors. I'm just wondering whether you just use the same threshold as a constant across your whole x-axis.
Yeah, it's a good question. So here we're only varying the thresholds within the range where we have enough patients, right, in the medical records to ensure that uniformly there's going to be a relatively low standard error, right? So if we change too much or extrapolate too much, right, then if I don't see any patients in my medical records, then my standard error is going to be very large. So then we don't want to make recommendations in that regime. Yes.
So maybe you have already explained it and I missed it. So my understanding is that by making this in silico, some of the patients that would have been excluded are added to this clinical trial. But I'm presuming there should be some sort of a sweet spot that as you increase your threshold, although there are more patients are added to your clinical trial, but eventually, because these are like you're deviating from ideal patients, the chance of this clinical trial being successful reduces, right?
That's right.
Spot. Do you have that?
That's right. Yeah. So I think that's also related to the previous question. So we have sort of a range where we are able to relax the thresholds, right, or do this design. If we go outside of that range, then either we don't have enough data to make a data-driven recommendation for the trials, or we actually do see that the trial results could get worse because of more adverse events.
First of all, great that you're getting so many questions in the middle.
Yeah.
Is there you're also assuming that there's the information for like the reason why someone got the drug, is encoded in the record, so you can actually do the thing. Is there a way to measure that? Have you thought about that? If you propensity scores going down, you still might have some kind of, because they're not randomized.
That's right. So, so we do compare the results from actual randomized things. Maybe I'll just, in the interest of time, explain the key idea here. So the idea here is that the way that we can get some level of randomization for validation is using natural experiments. So it turned out that if I look at 5 different pharma companies, the same checkpoint inhibitor, similar drugs, right? So they actually would use different thresholds themselves, right? Maybe some would use 1, other would use 2, right? Just because of sort of randomness of which teams are designing that trial. So we're assuming basically using that as essentially like an instrument to say that, okay, so how would the results of that natural variation in the randomized clinical trials compare with the results we get from the observational data? Right? So they do turn out to be very highly consistent. So that's how you can get some additional validation. Okay. Maybe one last one, and then I'll move on to it. Yes.
So what is exactly the hazard ratio? So what's the event that you're
Yeah. Thanks for the question. So the hazard ratios here, in this case, is for overall survival, right? So basically death. So you can also measure hazard ratio, and we have done that for time to progression, right? Which is how long, when does the patient move on to the next line of treatment, and you get similar results for that. Okay, so just to summarize this part, right? Hopefully this gives people a sense of how we can leverage these ideas about these in silico trials to help us make data-driven recommendations to make actual clinical trials maybe more efficient and more diverse and inclusive, right? As many of people have pointed out, as we discussed, right? So these are observational data, so we do try to use data from actual randomized clinical trials to validate them, and also use data from other cohorts, right? So we use data from other hospitals beyond Flatiron to validate these results. While I focus on cancer here, I think similar methodologies and challenges can be applied to other diseases, and we're looking at a few other diseases. So we're very excited that this tool is now actually being used by the pharma industry, and it was actually recognized as the top data science innovation by the pharma industry last year. Okay, so more briefly, for the second part, I just want to show you how you can take similar ideas, actually the same idea of these clinical trials that use that to discover predictive biomarkers, right? So this is something I'm actually even more excited about. So it turns out that we have these medical records for these patients from Flatiron, and also for a large number of these patients, many of them had undergone foundation medicine testing, which basically is an NGS-based test to measure their tumor mutation profiles, mostly before the first-line treatment. Right, so we have this for over 40,000 patients across a variety of cancer types. Right. And what I'm interested in doing with this is basically look for, are there particular genomic alterations, right, before the patient started treatment that can predict whether the patient is going to respond to that particular immunotherapy or targeted therapy or chemotherapy. Right. So that's sort of really the holy grail of precision medicine. If you can do that, then you can maybe help with treatment recommendations. And sort of similar setups before. So I won't. Yeah. So I think it's a very similar conceptual setup, right? So now let's say if I'm interested in looking for all these bladder cancer patients who took immunotherapy, right? So I can go into my medical records, find all of those patients, and I'm interested in maybe a particular tumor suppressor, right? This gene APC, right? So I can look for, okay, among all of these patients in my medical records, which of them had mutations in APC while given immunotherapy. I create one digital cohort. I can also create another digital cohort of ones that's given immunotherapy, bladder cancer, but did not have mutation in this gene APC. Right. So in these two cases, I can still compare them, right? Match them, compare them, and see, okay, does having mutation in APC correspond to a better survival outcome for those patients? So in this particular case, right? No, we did find that the patients, that's the orange curve are the patients that had APC mutation, and then they were given immunotherapy, right? Compared to the blue group of patients who did not have APC mutations, the y-axis or overall survival, we do find that patients that had APC mutation had much better response to immunotherapy, right? And similarly, you can ask, okay, so is immunotherapy really the best treatment to give to those patients that have APC mutation and bladder cancer? So we also find that these patients that had APC mutations, they do respond much better to immunotherapy than to any other treatment that's available. Right, so that actually combined information corresponds to evidence to suggest that there is a positive interaction between this mutation in gene APC and immunotherapy, which is not really known previously. Right, and mathematically, it comes from looking for these interaction effects in the survival model. In this particular case, we're able to replicate this result using data from a randomized clinical trial. We're collaborating with our colleagues at Genentech and Roche. And Roche had actually run a bladder cancer trial testing atezolizumab, which is immunotherapy, right, over 900 patients. It's a randomized trial. They did mutation profiling using foundation medicine tests for all of those patients. Right? And then in this clinical trial, we validate our results and see that, okay, there is indeed a positive interaction between APC mutation and immunotherapy. So patients with APC mutation had much better survival outcomes. So the hazard ratio is very small here, which means that there's actually a huge effect size. So just one example for this APC gene, but we can use similar kinds of approaches. We actually identified over 450 similar kinds of strong mutation treatment interactions. Basically, each of these interactions corresponds to a strong predictive biomarker that indicates that if you have this mutation, this is sort of maybe the best treatment for you. Right, so this is a visualization summarizing some subset of these results. They're broken down by different cancer types and by different treatment classes within each cancer type. So I just colored these genes to be red if there's the negative interactions. That means that, for example, if you have EGFR mutations in lung cancer, then you have worse response to immunotherapy. But if you have a KRAS mutation in lung cancer, you have better response to immunotherapy. Right? So red means there's a negative interaction, blue means there's a positive interaction. The size of the gene is proportional to how many patients have those mutations. So I think the power of doing this in silico analysis is that previously in the literature, there were only a handful, maybe less than 60 genes that were actually known to have these strong interactions using human data. By doing through this in the medical records, we're able to expand that by order of magnitude. So this was in a recent paper that we just published a couple of weeks ago. I won't go into too much detail, but we can actually find patients with multiple mutations, right, concurrent mutation in their genome. And we also show that there's actually very strong mutation interactions. So if you have multiple mutations that actually affect your treatment response to targeted therapies. Right, so all of this, we actually created this website called precisioncancer.org that has all of these results because there's a lot of these findings that people can hopefully this will be a useful resource to folks. Any questions about this part? Yeah. Yeah, so we checked for like tumor mutation burden. Oftentimes, I think TMB has a relatively, in our data, has a pretty marginal effect on, for example, immunotherapy outcomes, right? So even if we include that as a covariate, the effect of tumor mutation burden is actually often much weaker than the effects of, for example, APC of these other specific mutations that we saw. Okay, so maybe just to wrap up and just summarize maybe a couple of the main lessons that we learned from running these in silico trials. Right, it's the same kind of algorithm that I used behind both of these research projects, right? And the first one is what we already mentioned is that often the biggest factor that determines whether these in silico trials are successful is whether we're able to use different covariates to control for the potential confounders and biases, right, in these digital cohorts. So that can be done either using propensity methods or it can be done using other machine learning methods. But the key is really like, do we have actually enough covariates to be able to really control for a lot of these confounders? Right, in our particular experience, right, so here are some of the key covariates that we need to include, right, in order to control for these factors. So certainly demographics and line of treatment is really important, right? Line of treatment is critical because the early lines means the patients are healthier, later lines means they're more severe, right? Something that's really important is like ECOG, which measures like the overall health status of the patients. Right? It's noisy, but it's quite important. We had found like time of diagnosis, which we can extract from EHR to also be quite critical, especially the time gaps, right, between diagnosis and treatment. So that's something really important to account for. And surprisingly, maybe another important thing to account for is like insurance status, which is often, you can extract that from Flatiron, and it's also sort of a good proxy for sort of a variety of socioeconomic factors. What we often try to do is to only look at patients that have sufficient long follow-ups, right? So basically, if I only seen them in the medical records a few times, then maybe I don't know if I have very good complete data from those patients, right? So we often have to filter out those patients. There are a bunch of, I think, useful robustness checks and external validations that we recommend people to do. Right. So one thing with robustness checks is that you can, after you try to use all of your covariates, you can actually see, are your digital cohorts actually well controlled? Right. So these can be tested based on these balance tests. Right. So here's just an example of this. For example, in lung cancer, we show that after our computational matching, right? So that's the orange curves, right? These standardized mean differences between the digital cohorts are very close to zero. So that means they're well matched after these adjustments. We try to, as much as possible, we recommend using data from orthogonal sources to validate the results. So maybe an interesting note is that most of the data from Flatiron come from community clinics, right? Over 80% of the patients come from community clinics. So to validate these results, we actually use data from academic centers, which often had very different distribution of patients and distribution of treatments, right? And the results are consistent, so that gives us, that gives quite a lot of robustness to the results. And certainly, as we saw, I think we try to compare the results with randomized trials as much as possible as sort of additional validations. So just to summarize and to wrap up, so here the key message is to hope to give you a sense of how you can use this idea of in silico clinical trials, which leverage the data from these very rich medical records and clinical genomics information, right, to computationally emulate trials, right? And you can use this as a way to help us to design these very expensive trials, but also use this to discover new biomarkers. I think there are a lot of other applications we're excited about, including label expansions, et cetera. So here are the papers that describe this work in more detail. And to acknowledge again the collaborators, Rishan, Shimmer, and Ryan, and our collaborators at Genentech and Stanford. So happy to take more questions.